Hello, my name is Gonzalo, and today I'm going to present to you my science fair project, Antibiotic Resistance and Immune Hope. When you have a bacterial infection, what does your doctor prescribe? Antibiotics, right? Antibiotics kill most of the bacteria. However, there are a few that survive because the antibiotic didn't get to them or the bacteria just become resistant to antibiotics. Even though it is unlikely for a bacterial cell to develop an antibiotic resistance trait, in a bacterial infection, there are billions of cells, and this makes antibiotic resistance much more likely to happen. Multi-drug resistant infections have high mortality rates and high financial costs. Essential oils are oils containing volatile aromatic compounds from plants. There is evidence that some herb-based essential oils have antibacterial properties. Essential oils have been tested against multi-drug resistant strains and human pathogens. In our previous project, we established the antibacterial properties of the essential oils of thyme and oregano against Streptomycin resistant bacteria found in apple trees. We have extended this study to test for the antibacterial properties of these essential oils against E. coli that is resistant to Streptomycin, chloramphenicol, and penicillin. We chose these three antibiotics because they have different mechanisms of action. So this brings us to our hypothesis. We hypothesize that the essential oils of thyme and oregano will inhibit the growth of bacteria that is resistant to Streptomycin, chloramphenicol, and penicillin. In order to test for the antibacterial properties of our two essential oils, we used the disc diffusion method. The disc diffusion method consists of soaking the sterile paper disc in the substance you want to test, and then adding them in a lawn of bacteria. The antimicrobial activity of the substance is then measured through the diameter of the inhibition zone after it has been left to incubate for 24 hours at 37 degrees Celsius. Two full teal dilutions of each oil up to 1 to 64 were tested for each condition, and we used ethanol as our diluent. A total of 6 agar cultures with the selective antibiotic and 5 agar cultures without the antibiotic were analyzed for conditions. We wanted to confirm that the selective pressure created by the antibiotic didn't affect our results. The antibiotic kanamycin was used as our positive control, and ethanol, our diluent, was used as our negative control. The results are summarized in the graphs below. In these graphs, I plotted the size of the incubation zone in millimeters versus the two controls and the dilutions of the essential oils of thyme and oregano, respectively. The bar graph represents the mean, plus and minus the standard deviation. The statistical analysis included an ANOVA with donuts post-hoc. To keep this short, I'm going to focus on the results from the Streptomycin-resistant bacteria. The green or blue bars with a plus sign represent the results of cultures where the selective antibiotic was present in the agar. The gray bars with the negative sign are conditions when there was an absence of selective antibiotic in the agar. As expected, in our negative control, ethanol, the incubation zone is 0, and for the positive control, kanamycin, the incubation zone averages 25 millimeters. As you can see, as the concentration of the oil is reduced, the diameter of the incubation zone is reduced as well. The pound signs above the bar graphs represent when P is less than 0.0001 when compared to ethanol. We had similar results when we tested the essential oil of oregano. Overall, the essential oils of thyme and oregano inhibited the growth of bacteria resistant to Streptomycin, chloramphenicol, and penicillin in a concentration-dependent manner. There was no significant difference when we compared the culture with and without the selective antibiotic in the agar. Next, we asked the question, do antibiotic-resistant bacteria develop a resistance to the essential oils of thyme and oregano? To test this, we left the agar plates in the incubator for a minimum of 10 days. In the picture on the left, there is a culture after 24 hours of incubation. The arrow highlights the zone of incubation in the dilution of 1 to 8. On the right, there is the same plate after 10 days of incubation. The arrow points to the growth of a colony inside of the incubation zone. Based on these novel results, we believe that antibiotic-resistant bacteria may also develop resistance to essential oils. Next, to summarize my results, the essential oils of thyme and oregano inhibited the growth of E. coli resistant to Streptomycin, penicillin, and chloramphenicol. The combination of the selective antibiotics and essential oil did not influence the inhibitory activity of the essential oils of thyme and oregano. After several days, colonies grew into the inhibitory zones for both thyme and oregano oils. In conclusion, essential oils, such as the essential oils of thyme and oregano, have antibacterial properties against bacteria resistant to Streptomycin, chloramphenicol, and penicillin. Therefore, the essential oils of thyme and oregano may be an effective treatment for antibiotic-resistant infections. My future directions are to test the essential oils of thyme and oregano on bacteria resistant to other classes of antibiotics, such as macrolides and tetracyclines, and to confirm whether satellite colonies have developed a resistance to the essential oil of thyme and oregano. If the colonies are truly resistant, I could sequence their genome to look for mutations.